Hello, everyone. Welcome to today's live broadcast, Challenges in Biosimilar Bioanalytical Assays and Sample Processing. I'm Mike Christel, Managing Editor of Applied Clinical Trials, and I'll be your moderator for today's event. We are pleased to bring you this web seminar presented by ACT and sponsored by Celerion. Celerion, a global clinical research organization, offers a unique combination of medical expertise, clinical operations experience, and scientific excellence that gives its clients the confidence to make fast, accurate decisions about their drug development path. Its services include comprehensive clinical development services from phase 1 through 3, including patient dose response studies, cardiovascular safety, and product labeling studies. In addition, Celerion offers statistics, data management, and biostatistics and bioanalytical services. Its founding mission is to help clients get their drugs to market quickly so that they can touch the lives of family members, friends, and people in need around the world. We first have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You may also locate the resource list provided by the sponsor on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. Should you have any technical problems viewing or hearing this presentation, please click on the question mark Help widget in the dock at the bottom of your presentation window. I would now like to introduce today's speaker. We are pleased to be joined by René Wuttke. René is a Bioanalytical Principal Investigator at Celerion, Switzerland. He joined Celerion to support ligand-binding bioanalytical studies on pharmacokinetics and immunogenicity assessment. He has been leading recent analysis of clinical biosimilar studies employing highly automated assays. René studied biochemistry at the University of Potsdam, and he holds a PhD in physical biochemistry from the University of Zürich. Thank you for joining us today. René, please do get us started.
Hello, everyone. Welcome to this webinar. Today, I want to talk to you about an often overlooked fact or overlooked part in biosimilar development, and that is bioanalytics, which we are providing as part of our services. So today, I want to talk about the specific challenges in the bioanalytical assay and sample processing for biosimilars. So, for biosimilars, there are several challenges we have to deal with, and we can help kind of only with a subset of this. Because after your clinical study, of course, you want to have most identical results, most identical PK curves, most identical safety profiles, and we want to help you achieve that. But there is only a certain subject we can help with. When we are first talking about PK studies, that is for most people the most important part, the pharmacokinetic profile, having it similar between or identical between the biosimilar and the innovator. There are 4 main contributors to variance between those. And that is a normal subject-to-subject variability, so the biological variability. And then there's also a lot-to-lot variability in the drug concentration of both the biosimilar and the innovator. And of course, there could be, of course it is not desired, remaining chemical differences between the biosimilar and the innovator, which then also are picked up by the assay. And major complication, which is not really specific to biosimilars, but simply for doing work on biologics, is the relatively low precision and accuracy of ligand binding assays compared to other methods like biophysical methods. And there's really only 2 things we can work with, so these are the last 2. And I'm going to talk to you why. So what we cannot help with is really the subject-to-subject variability. That is simply the biological variability you have to deal with. But we can, of course, help you in generating your your clinical trial design in a way that this minimizes this. Helping you with that are my colleagues, for example, in Montreal. There's very little we can do for lot changes in the concentration of your material, but we can sometimes characterize any differences and be sure that this is a difference coming from there. What we can certainly help with is actual chemical differences between the biosimilar and the innovator, if they exist. We will be able to characterize if they are present, and this will inform us on how to further proceed in our bioanalytical strategy. Mostly this means choosing between a 1 or 2 assay approach, so having a certain single assay for detecting both the biosimilar and the innovator, or using 2 assays for detecting either one or the other. And also, of course, with the precision of the ligand binding assay. There are some things, little things I will show you later, where we can push really the performance of the assay. And we have developed a 2-stage process on setting up assays in the analytical laboratory. And for most of the cases, this is a single assay. So that is nothing really, nothing new. There is an industry consensus that it is an ideal situation that both the biosimilar and the reference material, the innovator, should be detected by an identical assay, with a preference that this assay consists mostly of the biosimilar and the reference product is measured essentially in a biosimilar assay. So that means the assay components should detect primarily the biosimilar, and then you make the scientific exercise of showing that this assay actually can detect the innovator just as well. And that is what we are meaning within One-Assay setup. And to show that these are the same thing basically, or they are detecting the same thing with equal performance, we have a 2-step approach. And first, in the development phase, in our R&D lab, we establish that calibration curves between these components are overlapping essentially. Ideally, you want to have them overlapping. It is probably workable that they are parallel, but that is not ideal. And then later on, in the validation phase, we again confirm this comparable reactivity, then not on the level of the calibration curves, but by a systematic comparison of quality controls against a calibration curve of the biosimilar. And we also basically do a double validation. All important parameters you have in a bioanalytical method validation are again repeated for both compounds. So again, let me be clear that bioanalytical similarity is not chemical similarity. Of course, you usually have a wide array of biophysical methods to characterize your proteins. This is, of course, something we cannot really do. We have a detection antibody and a capture antibody, and they're essentially only reporting on that moiety where they are binding. Of course, we can try several ones, but there's a clear distinction between the types of similarity. Again, I said we are comparing standard curves, and we ideally want to have perfectly overlapping curves. When we don't have overlapping curves, but they are basically shifted on the y-axis, on the signal axis essentially, that is usually an indication for that there is a difference in concentration between the biosimilar and the innovator. And that is something we sometimes pick up even as a second step. For example, we had the case that there was a lot of adsorption of the biosimilar against tubing and other containers because it did use slightly different formulations, and that showed up as a concentration difference. If you have non-parallel curves, that calls for two-assay approach. That is really, really rarely the case. Most of the normal new biosimilars, they actually can be detected with a one-assay approach. But how do we evaluate this and how we do the test for that? And this is actually quite simple. So this is an approach which has been formulated by the AAPS working group for biosimilars and has been published a few years ago in a paper by Marini and et al. And we start off with producing several plates. So this is a graphical representation of a 96-well plate, which is what usually is used with ELISA. And we have differently colored simply the standard curves and of the biosimilar and the innovator. It doesn't really matter which one is which. We are doing double determination. That's why there are 2 columns for every data set, so to speak. And then we have 3 data sets from the innovator and 3 data sets from the biosimilar, and then we do that again 3 times, 3 plates. So we have 9 data sets each, and we basically average those and comparing those. And how does this look like? So that's here then from such an experiment. There we have 3 curves which correspond to the 3 plates. And now you really want to be relatively critical about how the collaboration curves are shaped. This data is from an actual biosimilar versus innovative comparison, which was done in our lab and also later received positive approval by the authorities. And that is really the ideal case here, where you have perfectly overlapping curves and there's nothing really you have to second guess about them, because if they are not really overlapping, you have to define essentially some criteria on how to determine the differences or how to quantify the differences. If they are clearly overlapping, that is usually not the case, which is preferable, certainly, because you don't have to do any fancy math, essentially. So in the second step, we essentially move away from the non-GLP development phase of the project and move to the GLP lab validation. And we want to basically stress the assay now as much as possible in the validation lab. Usually most people will not tell you that. Most people want to keep everything nice and tidy. We also keep it nice and tidy, but we intentionally introduce a variation by changing up incubation times, incubation temperatures and things like that. We want to really stress the assay now and basically show that despite treating it so badly, we will still achieve all acceptance criteria which are demanded by the authorities. So basically what we are doing here is we are making different independent dilutions from stock solutions to the levels of QC levels now. This is also usually not necessary when we're talking about the normal ELISA assay validation, where this is usually a single dilution from the stock solution. But we do it now multiple times and we average out the effects. So we are basically adding a lot of variability from just the preparation in there. And then we basically measure the QCs of both compounds again on a plate. So that looks very similar to what you have seen before. Now, of course, we have made the decision that we move to a single assay and we have only one standard curve on the plate, which is ideally from the biosimilar. And then we put on 5 different levels, so LLOQ to ULOQ, that basically means different levels of QCs spread out over the whole analytical range, again from the biosimilar and innovator. And we also shuffle up in the 6 different runs we are doing the location of each of those sets. But they are coming from 6 different dilutions, which is usually not the case when you look at the normal validation. And when we have gathered all the data from this one, we are basically analyzing them according also to the AAPS white paper about biosimilars, and this are a lot of numbers and a lot of graphs here. But what is really important is essentially we're looking at the bias between the curves, and we look if this is not above 10% or actually 20%. So in this case, this is a green line here on the left plot, which goes from the ULQC to the LLQC, so from high concentrations to low concentrations. And you see here that the difference is usually not much greater than maybe like 5% or so. That is a typical picture. And we also check if this error here, this error bar, is not more than 30%, 35% at the ULQ. So that is a standard, basically, evaluation of the data according to the Marini paper, which has gathered a lot of industry acceptance. And of course, then we do the relatively boring stuff of doing the selectivity of the assay, doing the dilutionality of the assay, doing the stability of the assay. And I think what sets us apart from many other companies, we always do this with both compounds, because what we have gathered from the responses from the authorities is that they really want to see all this information doubled up, even if this is often a relatively simple exercise with not much deviation. Well, this is what you expected. There's not much deviation. And there's one thing when you, of course, now have validated your assay and you're moving away from the assay validation and into your study and into your clinical sample analysis, then you want to employ a few tricks. And for plate-based assays, this has a lot to do with how you essentially place or plan your daily work on the biosimilar and innovator samples. And we are usually blinded, so we have to basically do everything in a symmetrical way, so to speak. For example, if you imagine a PK profile as shown here on the left, with some fantasy numbers, so to speak, you of course have a fast increase in concentration up to the Cmax and then decay. And of course, you are not really interested in that the PK profile is really shown perfectly. You're really only interested in the difference between those. So that means, for example, when you have 96 volts available minus, of course, all the QCs, and you end up with 15 or 16 slots, so to speak. You want to put half of those from the biosimilar and half of this from the innovator at corresponding time points. And of course, because usually in antibodies you have a lot of dilutions, you want to optimize the dilutions so it hits the optimal spot of the calibration curve, which is like illustrated here on the right side. But I think what is the biggest contributor to variance here is the human factor on pipetting things. And we are in Switzerland, of course, manpower is very expensive, so how we are dealing with by basically getting rid of the human factor. And it should play now video exactly of our instruments where we are basically automated lots of the assays. And that has several advantages. The main advantage is just, of course, that there's always an even execution of the assay. And the even bigger advantage is that you can see here that all these samples are barcoded, and not only the samples are barcoded, also plates are barcoded, as you can see here. So there's always a tracking of all the steps done to a sample from the beginning to the end with the best performance. What you have seen in the beginning was basically our sample preparation robot, and just a few pictures here from our second robot, which basically does the actual execution of the ELISA assay. That is relatively simple. You basically have here a plate which goes through all the coating, binding, washing steps up to the section in our robotic system. And that is how we basically reduce precision and accuracy errors to a minimum, which gives you better comparison between your biosimilar and your innovator. Okay. So that is the first part of my talk. And I think there is a question now coming up for you.
Yes, yeah. Thanks, René. Yeah, thanks for that. Yes, so now we'd like to ask our audience to participate in a brief polling question. You can see it there on your screen. Please click directly on the screen to enter your answers. Here's a question. What is the greatest challenge you see with biosimilar development? And there you see your options there: production synthesis, clinical PK/PD, clinical efficacy, or regulatory interaction. Once again, what is the greatest challenge you see with biosimilar development? Give the audience a few seconds to respond. All right, great. I'd like to thank you for participating in our first poll, and we'll swing it back to René.
Thank you. So in the second part of the talk, I want to talk now about the slightly more messy issue of not about the PK profiles, but actually working with the immunogenicity of your product. And there's sadly much less consensus about of what experiments should be done to show that assays are detecting the immunogenicity of the biosimilar and the innovator in a comparable fashion. And this here is simply a literature list showing the different approaches. And you can see there is, in a relatively short amount of time for bioanalytical standards, several papers have been published which don't really reach to a consensus. But what I want to show you now is our basically also two-stage setup and how to show that, and that has been also been successful in submissions to European and American authorities. So here it is the one or two assay approach. Is of course has advantages and disadvantages. Two assays, of course, they remove any bias between the two compounds, but of course, that basically means you have to measure every sample twice. because we are blinded at that stage. So it basically doubles your determinations, and it makes it basically from a flow chart on how to proceed with a sample slightly more complicated. A one-assay approach usually generates false positives, but it removes also the bias between that. Again, a one-assay approach actually has been emerging as a consensus in the biosimilar community. Has not really been confirmed by the authorities so much, but also I have not seen any major objections. In this case, this means I actually wanted to talk about this again. So there is a significant lack of consensus. It's also not clear where to tackle that problem, at which stage of the bioanalytical assay development. And big confusion is also on where to put the results of this determination and the results of these experiments in an integrated summary of immunogenicity, for example. That is maybe possible. And we also contributed to those in the past. And what has basically crystallized for us now, which as a gold standard, is that we do two main experiments. And the main experiment, and that is why it's very important for us if you want to place a bioanalytical assay with us, that you come to us very early, because we usually start, or we usually recommend that you generate polyclonal antibodies against the biosimilar and the innovator in the same lab, and we will take care of that based on the same immunization schedule and so forth. So it is usually not okay to just work with polyclonal, with commercially available or with monoclonal antibodies. That is still the gold standard, to use polyclonal antibodies, which are known that they are binding the biosimilar. And that's why it makes sense to generate those beforehand. And that takes several months. So when we go ahead, then we have these two antibodies in the lab, and we basically produce both assays, meaning we use functionalized biosimilar and functionalized innovator as a capture and detection reagent, and it essentially gives us something very similar to the PK assay, and then we're also doing another experiment which I'll come to later. So how does it look like? Here is simply a graphical representation of what types of molecules we have here on the right side. So we have basically everything with the biosimilar on the very top. Let's say blue is the biosimilar. So that means that we are detecting the positive control generated against the biosimilar using capture and detection reagents which are functionalized using the biosimilar. And then we do the same with the positive control generated against the innovator. And basically this is how the two curves in the left plot are generated. This is an early data set. Usually we have much more data now. That makes it even look much prettier. So here these curves are parallel, which is sufficient. The difference between those only comes from the different labeling efficiency of the different compounds, so that is fine. And then we do this again for the innovator assay here, everything with the red blobs, and we test both generated positive controls. If you see this picture, then we are essentially okay. We know we can use a single assay for the detection of both antibodies. And there's one additional step we are doing during the validation. That is basically we're doing pretty much an expanded free drug tolerance experiment, where we take the then positive control, so the anti-biosimilar positive control, and we titrate it with both the innovator and the biosimilar, and then look how parallel or overlapping these curves are. So on the left, there's an example from Riding et al., who popularized this approach. And this is on the our right-hand side. This is also real data from biosimilars. And we, depending on how complicated this is, we basically also add more points and more levels to this. We basically look at these overlapping. And if both are okay, then we basically are fine with using a single assay approach, using only the positive controls generated against the biosimilar as our controls. And we proceed then with normal validation. And as a last part, I just wanted to give you a little example, a little case study on one of our biosimilars we were working with, and that was Pegfilgrastim. And we supported of the 4, I think, successful Pegfilgrastim biosimilars which are on the market now in the European Union. We supported 2 of those, and we recommended, of course, very similar bioanalytical methods to both companies. But we opted to validate everything again for both companies, so that they could be sure that their compound is really performing as they expected in the bioanalytical assay. And there's a little bit to talk about the Pegfilgrastim. I think the major complication here is that this molecule is pegylated, and at least the EMA guidelines from Europe still for the characterization of the binding moiety, which can be a source of the similarity. So they basically want you to look what impact the pegylation has on the product. That is usually not a big problem for PK assays. It's basically behaving as any other molecule in the PK assay. As we expect, we just use capture and detection antibodies against the protein body, but the problem is now that for the ADA assay, this is very much not the case. They really have to deal with pre-existing anti-PEG antibodies, which every one of us has. That has many different who blame cosmetics or other things which are in plastics, and they're getting into our body, and we are generating antibodies against it. And depending on the literature sources and also from our experiments, we can see that from 5 to up to 30% of the population, people are having pre-existing anti-PEG antibodies in their bloodstream. And if you basically apply all the typical pegylation methods and procedures for antibody assay validation to these compounds, you will get disruption, essentially. These numbers will not match up, and we circumvented that using certain calculations. We also tested really in a field approach where which binding moiety is used here, or the antibodies on which binding moiety they are binding. So that means that most all samples go on a screening assay first, and 30% are approximately positive. So actually, Pegfilgrastim is a relatively not immunogenic compound. So one would expect that most of the antibodies are against PEG, but we confirm that. And we basically use the four-pronged confirmation assay, which we called confirmation for the biosimilar and the innovator, which is called Nalasta. This is a normal confirmatory assay. And then we again added PEG or filgrastim. So filgrastim is just a protein body without the PEG in a confirmatory assay. And basically only if any of those positive in this confirmatory assay, then you would determine the titer, and you also would go on to a cell-based assay, a proliferation-based assay, which determines if the antibody is neutralizing. And of course, because we use PEG here in this previous step, we have to also add an anti-PEG control, a commercially available one, and this anti-PEG control that needed to be also very much characterized as suggested by the EMA. We did some experiments for characterizing this, for example, the Celerion, so that they requested us to characterize the anti-PEG control, and which is not shown here because I can't really show it. They also wanted to test us to different PEGs and things like that. So really characterize this anti-PEG control, which was a little bit surprising for us because it was only just a performance measure. It has nothing really to do with the assay. But in the end, it was very successful, and we could show that the spec is actually behaving as we expected, and would support the successful submission of our two clients, and they are now on the market. So, as a brief summary of the whole presentation, so I showed you how to characterize bioanalytical similarity, and we do that the first step in the method development, and then we really stress test this assumption, and we can use the same assay method validation, and then we really tighten up on the possible contributors to variance in the analysis, and we use that approach basically for the PK and also for immunogenicity assay, and have basically set up which in several biosimilar projects that they can give you a positive submission. At least it's not all. So that would be for me, and I would like to thank you for your attention.
Thank you, René. Thanks for that presentation. We did have a final polling question that we would like to ask our audience to participate in. It should be on our screen momentarily. All right, here's our final polling question. What are the services you are most likely to outsource? A, production synthesis, B, non-clinical in vitro or in vivo, C, clinical PK/PD, D, clinical efficacy, or E, regulatory. Once again, the question, what are the services you are most likely to outsource? And then you see the options there on the screen: production synthesis and non-clinical, clinical PK/PD, clinical efficacy, or regulatory. Give the audience a few seconds to input their answers. Thanks, René, for such an informative presentation. Before we get started on the Q&A session, I would like to remind our audience how to submit questions. You could submit them by typing them in the Q&A box, which can be found on the right-hand side of your presentation window. Okay, here's our first question from the audience. What is the difference between currently tested biosimilars and a general approach to the popular antibodies?
Well, thank you for the question. Thankfully, there's not such a big difference between generally large molecules and the popular monoclonal antibodies, which are basically the biologics of choice. As a bioanalytics group, we basically can process them very similarly. It doesn't really matter to us. Of course, they have some properties which need to take into account when designing the assays, but this is usually not the deal breaker. The general concepts on comparing assays in terms of biosimilarity are the same, so you basically can follow that pattern. From our personal experience, we have experience with both compounds. As you've seen here, Pegfilgrastim, that's a pegylated cytokine. It's not an antibody, but we just as well have worked with many biosimilars, which are antibodies as well. So the general concepts apply here just as well.
Okay, thanks. Another question in. Is there an impact on ISR?
Yeah, so the question on ISR, you meaning incurred sample reproducibility?
Mm-hmm.
Yeah, there is some impact on the ISR, because what people are usually doing with ISR, that they are selecting a few samples which fall into certain concentration ranges within the analytical range, so something which is on the top 30% or on the bottom 30%. And what happens here is that there's potentially the problem that you could enrich essentially either biosimilar or innovator samples or signals from those samples into your ISR assessment in an unequal fashion. And that is generally not desired, especially if there actually is a certain difference in concentration between the innovator or the biosimilar. Either one of those has a higher concentration, might only be 5%. Then, of course, you have much higher concentration of samples from that compound in that selection of samples. So in that case, what I'm doing here to circumvent that is slightly deviate from a standard procedure, and first I analyze concentrations, concentration brackets at the low and the high end, and then I analyze the frequency of certain time points within the clinical protocol showing up in these concentration ranges, and from that I choose several time points from which I then select samples randomly from different patients or different subjects. That is, of course, because I'm blinded. I don't really know from which sample, from which treatment group they come. So, of course, there is an error here, but that is unavoidable because you're blinded. But in that way, you have an even distribution between ISR samples from your biosimilar and from your innovator.
Okay, another question in from the audience. What changes have become important for biosimilars in the 2018 and 2019 FDA guidances on method validation?
Yeah, so that is right. There have been 2 new guidance documents released by the FDA. One in 2018 on the PK method validation, and in 2009 on the ADA method validation. And thankfully, there's not so much really to be concerned about as a biosimilar developer here. They are mentioning biosimilars, but only in passing. There's no really separate section for them in a big way. But there certainly have been changes in general on the bioanalytical process or the bioanalytical method validation. And just after the release of the ADA guidance document in 2019, in January, we actually had in February FDA
audit in or FDA inspection in our facility here on a biosimilar project, where I also was the lead, and that came out quite well, if you can say that, for an FDA inspection. So there was no 483. The auditors were the inspectors were very satisfied. There are certainly new requirements in terms of the documentation process of the whole bioassay development, validation, and sample analysis procedure. So they are demanding much more, and they are also demanding more oversight over the study in terms of trending analysis and things like that. Here it is important that your method development part, for example, the biosimilar comparisons I just showed you in the talk, are properly documented and ready to be shown to inspectors. So they generally agreed with this concept here, not in all points, because not all of these points were actually relevant for that project, but so far there is a little extra to do, but there's much more to do compared to previous years.
Thanks, René. Unfortunately, we are out of time. Any questions we were unable to get to during the webcast, Celerion and René certainly will respond to you directly via email afterward. I want to thank the audience for attending and for participating in today's event. I would also like to thank our sponsor, Celerion, for making today's educational webcast possible. You will receive an email from Applied Clinical Trials alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thanks to all for joining. We will see you next time. Goodbye, everybody.